What do dead cows, rat poisons, and medicines to prevent strokes have in common? Every year we hear about a few new drugs getting approved to treat targeted diseases, called indications. What we don't hear about is that in the same year, thousands of candidates fail and never become drugs. Decades of record shows that only 2% of the drug candidates are approved and make it as a marketed drug product. The drugs that make it take in average 12 years and hundreds of millions of dollars to develop. This is one reason why drugs are expensive. Today I will tell you in simple terms how a drug is developed. The drug development process typically begins in a university laboratory. Academic researchers obtain grants from governments or institutions or pharmaceutical companies. Specialists want to understand what causes diseases at the level of our cells, or even smaller, at the level of molecules that make up parts of the cells. These studies aim to understand the molecular pathways that lead to diseases we want to treat. For example, diabetes, infections, and heart conditions. Understanding the pathway will help to identify the targets that need to be stimulated or blocked to treat a specific condition. When a target is identified, researchers then look for molecules or compounds that stimulate or block that target. Such molecules or compounds then become candidates for development into new drugs. Did you know that many of the medicines we have today come from natural compounds such as plants or marine animals? I'm going to tell you a story as an example. 100 years ago, a mysterious cow disease appeared in America. Ranchers did not do anything different, but cattle were bleeding to death. Cows bled to death after having their horns removed, and bulls bled to death after castration. An expert veterinarian named Frank Schofield solved the mystery. The killer was not a virus nor a bacteria. It was a plant with the cute name of sweet clover. Schofield noticed that the difference between healthy herds and herds that bled to death was the presence of rotten sweet clover. Schofield did an experiment on rabbits. He fed one rabbit healthy sweet clover and the other rotten sweet clover. The rabbit that ate the healthy sweet clover was fine, and the one that ate the spoiled clover died of a hemorrhagic disease. The word hemorrhagic is a combination of two words in Greek. Hem, which means blood, and rhagia, which means explosion or rupture. Schofield repeated his experiment and got the same results. For rabbits, lucky for rabbits, we have good control over animal experiments today. For Schofield, his job was done. He simply told ranchers not to let their cows eat spoiled sweet clover to prevent the blood disease. 20 years later, a chemist named Carl Pollink and his team set out to isolate and identify the hemorrhagic agent from the spoiled sweet clover. It took them 5 years to extract it and understand its molecular structure. We call this the molecular characterization part of the drug development process. Link learned many interesting things. Healthy plants have compounds called coumarin, which are completely harmless. But coumarin are attacked or metabolized by certain fungi and become different compound, warfarin. Bleeding can stop naturally when coagulation agents in the blood help form clots. However, warfarin acts as a strong anticoagulant to prevent clots, and that's why animals bleed to death. With this discovery, warfarin was repurposed as a poison to kill pests like rats. So now you know what dead cows and rat poison have in common. I will tell you next about how warfarin relates to a stroke prevention medicine. Link extracted warfarin and identified its chemical properties. We still didn't know how it worked until just 40 years ago. Today we know its mechanism of action, which is the way this compound works in the cells. Warfarin inhibits an enzyme called epoxide reductase, which in turn interferes with the metabolism of vitamin K, which works as an anticoagulant in blood. This is good news because we know what to do whenever we get a warfarin accidental overdose. We just used vitamin K as an antidote. You may be wondering, But Ara, couldn't we exploit warfarin anticoagulant properties and make it a therapeutic medicine out of it? If you're asking this question, you're already thinking like a scientist and a pharmaceutical professional. The answer is yes. In fact, warfarin was eventually developed successfully into a medicine to prevent strokes by mitigating clots in blood vessels, taking advantage of its anticoagulant characteristics. It was widely used for a time, but thanks to subsequent research, today we have anticoagulant drugs that are even safer with better efficacy. But Ara, how do you take a rat poison and successfully make it into a medicine for humans to prevent stroke? Now we come back to the heart of this talk, how a drug is developed. Starting with a characterized compound like warfarin, it must first successfully complete preclinical trials, then complete clinical trials before being approved as a drug. Let's begin with preclinical trials. Theoretically, the difference between something that is a rat poison and something that can be used as an anticoagulant medicine is the dose, or just how much you use. But how do we find out what dose is therapeutic and what is toxic? Unlike Schofield, today regulations prohibit us from simply feeding rabbits different doses to figure this out. The first step is in vitro studies experiments in labs that use cells in test tubes or boxes. Researchers perform initial toxicity tests not on live animals but on cells. The cells we use for this purpose are very special and deserve a separate dedicated video. Collecting in vivo toxicity data on cells helps researchers to design the next step, in vivo studies using living organisms to confirm safe doses and to understand how the compound is absorbed, distributed, metabolized, and excreted in a living organism. Unfortunately, developing a drug cannot avoid using animals at this stage. But the good news is that in fact, there are very strict industry regulations that govern the use of animals. Researchers need to obtain permission to initiate animal experiments. Researchers submit a very detailed request to explain and justify the purpose of the animal experiments. The request is reviewed by an ethics committee at the research center. The ethics committees are vigilant, and their mission is to ensure proper use of a live animal and to prevent cruelty and minimize suffering. To this end, the committee has the power to deny access or request changes to the animal experiment design. If the request is approved, researchers use animals from a place called vivarium that are bred specially for this purpose. They range from zebrafish, hamsters, rats and mice, and rabbits to larger mammals like pigs or dogs. Testing often begins on tiny animals and is only progressed to larger animals if researchers see signs that the compound has potential for therapeutic use. I have worked with animals in experimental labs. It was difficult to accept at first, but I found some peace when I saw how my mentor treated all her test animals with affection, care, and respect. She always strived to be as effective and accurate as possible to avoid repeating while making sure that no life was spent in vain. Most researchers have a profound love for life and care about lab animals. This is the monument to the laboratory mouse, which is in the Institute of Cytology and Genetics of the Russian Academy of Sciences, which commemorates the sacrifice of mice used to understand biological and physiological mechanisms for developing new drugs and curing diseases. Different animals are used depending on the disease being studied. This is because animals model body functions and diseases in different ways. For example, pigs replicate or model human infections quite well, so they're ideal for antibiotic studies. Rabbits, dogs, and monkeys all have human-like eyes, so they are used in ophthalmology. Guinea pigs are used for asthma research because they model our respiratory system well. Advances in science has also given us genetically modified animals that have certain genes turned off, called knockout, to model specific diseases. Knockout gene animal models are fascinating, and I will tell them about in another video. At the completion of clinical trials, both in vivo and in vitro tests, researchers understand a compound's positive effect, efficacy, and negative effects, toxicity, on living organisms, and have enough data to understand at what dose does the compound has a good efficacy and minimal toxicity. If at this point the researchers at the university believe that the compound shows good promise as a medicine, they need to take the compound into human clinical trials to confirm that the compound also has good efficacy and minimal toxicity in humans. But human clinical trials are much more expensive and cannot be carried out with only research grants. So typically, academic researchers will sell their rights to further develop their compound to large pharmaceutical companies with enough funds to do the clinical trials, in exchange for a series of payments and a share of profit, if the compound is successfully approved as a drug product. This makes sense. The researcher and their university receive payments for all their hard work, and they can use the funds for future projects. On the other hand, the pharmaceutical company obtains a promising candidate for a drug that may be approved. So how does this happen? Over the years, all countries have developed similar procedures for approving new drugs. All countries have established regulatory agencies to review drug approval. There is FDA in the US, EMA in the EU, CDE or NMPA in China, and COFEPRIS in Mexico. How do these agencies give approval? First and foremost, the agencies want to make sure that the drug is safe, that it does not kill patients or give them unwanted bad side effects. This is done in 3 phases. Phase 1 clinical trials must show that the compound is safe in humans. This can be achieved with a trial with a small number of healthy human subjects. Phase 2 clinical trials should show that the compound is safe, but also has efficacy in treating patients with the disease that the compound is intended to treat. This can be achieved with a trial with a small number of human subjects with the disease. Phase 3 clinical trial must show that the compound can have a meaningful efficacy in a large population on human subjects. This is done by a large clinical trial with hundreds, even thousands of patients with the targeted disease. What is meaningful efficacy? Well, if the disease already has an approved drug for the treatment, the trial needs to show that the compound is better, called superior to, the already approved drug, or that it works just as well as non-inferior to the already approved drug. If there is no approved drug for the disease, the trials can be designed to compare the compound with a placebo, that is, patients not given any real treatment. Some people may ask, why isn't the FDA or the EMA more active to do investigation on a new drug? This is a misunderstanding. Regulatory agencies are empowered to review clinical trial protocols and data generated from the trials, but they do not have the authority or funds to do research by themselves. At the end of the phase 3 trial, the agencies will review all the data. If the trials show that the compound has shown efficacy and safety versus a comparator drug or placebo, the compound will be approved as a new drug. I have tried to describe to you as simply as possible how a new drug is developed and approved. I hope I have conveyed to you how much super exhaustive work goes into making safe and effective medicines to help people with diseases and improve their quality of life. So let's make a summary. I have told you what dead cows, rat poison, and stroke prevention drugs have in common. A compound called Warfarin. One scientist identified rotten clover as the cause of cattle bleeding to death. Another scientist years later extracted and characterized Warfarin, the compound in rotten clover that was causing the deaths. This led to Warfarin being used as rat poison. More scientists later studied the molecular pathway and the mechanism of action of Warfarin as an anticoagulant. This led to Warfarin being developed as a medicine to prevent coagulation and stroke. This story is a nice example of scientists' contribution and the importance of scientific study to the improvement of human well-being. 2. Over the years, we have developed strict rules to develop new drugs. A new compound must go through rigorous tests in preclinical and clinical trials to prove its safety and efficacy before it can be approved and sold to the public. 3. The strict rules and protocols entail significant investment in time and money. From this, we can understand why some drugs are expensive. 4. There is nothing that is 100% safe and 0% risky because it really depends on its use and the dose in which it is consumed. Anyone who tells you otherwise is trying to sell you something. 5. Academic institutions, drug companies, and regulatory authorities have a common goal: to bring to patients new drugs that provide new or improved treatment for their diseases. They're really not driven by evil greed as many people would have you believe. So what do you think? Did you learn a little bit more about how medicines are made? If you think the video could have been better, please give me some constructive feedback. If you like what you have seen, please like and click subscribe and forward the link to friends and loved ones. See you next time.